Thanks, Dennis, and thanks, Jill, for the work behind the curtain. This is awesome. It's a really good turnout. I like the format of 4 o'clock. It gets more people in here. I hope you liked today's lecture. And I changed the title slightly. It used to say Harmful Algal Bloom Dynamics in the Southeast Florida region in the IRL. I'm going to talk about South Florida in general because I think it'll be more instructive, and it's of interest to all of us. Before I start, I did this at pretty much every lecture I've given here. There are so many people I need to thank that have helped me out, informed me about the lagoon, and just have generally got my science going. And I can't go through everyone on this list, but I thank you. And I especially thank the Harbor Branch Foundation for the support of bringing me here as an employee, essentially. They help support us with startup funds, and also for supporting my research directly. So, I had no idea when I came here what was going to happen. I study harmful algal blooms. As soon as the local media found out that I study harmful algal blooms, and thanks to our communications office, when we had some of the events that I'm sure you've all heard about over the last few years, I got a lot of calls. And I was on TV a real lot. It was a media blitz. And it was, at one point during a 2-week period, I was on the Today Show, the Weather Channel, the Nightly News with Lester Hull. I mean, it was just, and I got to spend the day with Gabe Gutierrez. If you ever watch NBC News and see disasters, they always send Gabe there. So it was quite, we got to see the sausage being made of how they produce media things. And, you talk to them for 3 hours and they use 10 seconds. And that's pretty much the way it goes. But like I said, I mean, there was a 2-week period where I literally was on TV or in print media every single day. And at first when it happens, you tell your family, wow, I'm going to be on TV tonight. By the end of 2 weeks, they're like, really? I don't care. So eventually my wife got really tired of it. And she declared me Dr. Doom of the algae bloom. That, it's a name that has stuck with me now with my family and friends. That, oh, you're on TV again doing that. You're depressing the hell out of everyone. And that, but it's an important point. I'm going to talk about something that, in the title, it's harmful. It's harmful to wildlife. It's potentially harmful to all of us. So while I want to joke about it, it's a very serious thing. And I don't want to scare you, but I do want to scare you. I want you to take this seriously and think about how you can possibly help or be more involved. So I'm going to talk about harmful algal blooms, and obviously I have to tell you what they are. I'm sure everyone probably knows, but just in case they don't. Harmful algal blooms are simply, or we call them HABs for short, they're concentrations or occurrences of phytoplankton that cause negative ecosystem or human health impacts. It's that simple. And they're occurrences of phytoplankton, so that brings up the question, what are phytoplankton? And these are simply microscopic plants. They're commonly called algae, and they include photosynthetic bacteria. And here's an example of a harmful algal bloom organism. But you can see this is not just a plain plant. It can swim. So this is why we also tend to call them protists, because they're kind of in between animals and plants. But they are plants. They have chloroplasts, and they conduct photosynthesis like your normal houseplant. And here's an example of one of these cells, and this is a scale bar. 50 microns is about the diameter of your hair. So they're truly microscopic, and some of them can be incredibly small. So how are they harmful to us? Well, there's a number of ways. Most people just think about this. They think about toxins. These things produce things that can hurt you. And these are some of the toxins that occur or can occur in the Indian River Lagoon. But there's also 3 other ways: high biomass, mechanical damage, and economic effects. And I want to go through each one of these and just give you an example of how it works. So I'll start with toxins. There are a lot of different toxins that algae can produce. There's toxins that can produce paralytic shellfish poisoning, diuretic shellfish poisoning, amnesic shellfish poisoning, neurotoxic shellfish poisoning, ciguatera, which is fairly common. And the way they can impact you is through a lot of different vectors or methods they can get into you. You can get contaminated drinking water for freshwater species. They can get into our water sources, and you can ingest toxins that way. And wildlife also can get exposed that way. A lot of algae, especially when they get to high biomass, they emit noxious fumes. And these fumes can actually contain toxin and cause problems. The Florida West Coast, the Florida red tide, which almost everyone probably knows about, that causes respiratory distress from you breathing it in, from the fumes it emits. Obviously, you can eat contaminated fish or shellfish, those things that feed on phytoplankton, which are toxic, that toxic then gets into them. So if you eat a fish that has algae toxins in it, you're going to get sick, potentially. And there's also recreational exposure. I mean, if you're just swimming around in the water that happens to have an algae, you might ingest it or get it into you. Or if your pet, your dog is swimming around in the water, you can just get an exposure by playing around the water. And there's two ways that these toxins can actually get you. I mean, most people would be familiar with this. This is an acute impact where you eat a fish or you get exposed to a high amount of toxin and you get sick immediately. And you know right away something happened. And it's sometimes severe response to it. But just as bad are these chronic impacts. So it's not necessarily, you might not even know it for 10 years that this chronic low-level exposure is starting to do something to you. And I'm going to talk a little bit about that later. So next is the high biomass. This is another way they can be harmful. And this can cause hypoxia, anoxia, and shading of the organisms. So here's an example from 2016 when we had the brown tide up in the northern IRL in the Mosquito Lagoon. And this is a picture from the St. Lucie Estuary. And in both cases, you can say, okay, there's a lot of that stuff there. It's pretty obvious. So what does that do? Well, there's so much of it there that the light cannot propagate down to the bottom. So if you have seagrass or plants that make a living photosynthesizing on the bottom of the lagoon or on the ocean or wherever, and all that algae at high biomass is there, the light is never going to reach them. So what's going to happen? This. So no light, no growth. If you take your house plant and put it in a dark room for a week, it's going to look like that. Well, that's the equivalent of what's happening here. The other one is this simple cartoon shows where you get just this very large biomass. In this case, this cartoon shows that you can have nitrogen loading, or it could be any kind of nutrient, it doesn't have to be nitrogen, that goes in and it essentially fertilizes the water and it creates a high biomass. A lot of things can grow, just like fertilizing your grass or fertilizing a plant. And eventually this algal bloom will die and it'll start to sink and decompose. In the process of decomposition, it's eaten by bacteria, and they need to respire like us. They use oxygen, so it uses up the oxygen in the water. If it doesn't mix fast enough, that oxygen disappears, and you get low or no oxygen. The animals that live there are either stressed out or they die. So I simply put it there. This high biomass in the bacterial consumption creates hypoxia, which is low oxygen, or anoxia, which is no oxygen. Nothing can survive that. How many people here have heard of dead zones? Oh, a lot. Unfortunately, it is not this. I wish it was. It is this. For those of you who have not heard of them. This is that cartoon I just showed you. This is exactly what goes on. We have the Mississippi River plume, which comes out. It carries a lot of nutrients from its giant watershed from farming and agricultural purposes and human waste and whatever else gets in there. It gets out into the Gulf of Mexico. It promotes a very large bloom, which eventually settles down to the bottom. And the decomposition and bacterial respiration eats up all the oxygen. That creates a massive dead zone. And this is an example of how large the dead zone currently is, or at least was on that date, in the Gulf of Mexico. That's many Rhode Islands in there, which is our always used. And it's been found through research they're doing that the dead zone can expand and contract, but it's actually getting larger and larger. So it's a serious problem. But it's just not there. These dead zones and these hypoxic and noxic areas, they're spreading around the world. So here's a map of these, what they call suffocated spots, and you can see a whole bunch of these around pretty much everywhere. And it's a problem that is probably being fed somewhat by, whether you believe in climate change or not, some type of global warming. As the oceans warm, they can hold less oxygen, so that's going to exacerbate the problem. And the more we have eutrophication and nutrient runoff into our oceans, the more we're promoting these blooms and making this happen. So this is not just constricted to the Gulf of Mexico. It's happening all over the world. And when you have these dead zones, this is what the normal repercussion is. You have massive fish kills. This routinely happens. You go down to the bottom and all the marine life is dead. Coral reefs can die. It's a very sad situation. And you can have a dead zone, but dead zones don't have to be something that's permanent like the Gulf of Mexico dead zone. Not permanent, but very persistent like the Gulf of Mexico dead zone, which that has lasted for years. You can have these anoxic and hypoxic events happen over very short timescales, just over a few days. So this is what happened during the brown tide 2 years ago. I'm sure a lot of you remember this. One night, there was enough biomass and enough respiration that it went very, very low oxygen and millions of fish died in 2016. So you can get these quick events that are just as bad for the ecosystem. We'll move on to the next one. This is mechanical damage, and this can be caused by mucus and serrations on phytoplankton, and I'll show you an example of this. This is an algae. It's a diatom. It's not a dinoflagellate. It doesn't swim. And it actually has a shell that's made of silica glass. So this is it has basically a glass container, and that's what it grows in. And you can see these very sharp serrations on these setae that grow out of its cell. So when these setae, when these get pumped into fish, they can get into their gills and essentially cut them up. And they can start to bleed, and it can kill the fish. So there have been out in the Pacific Northwest very large salmon kills due to blooms of this particular diatom. It's in the genus Cattosaurus. There's also this, which I classify under mechanical damage because it is a mechanical effect, but I'll use the brown tide up in the northern IRL as an example of an algae that can do this. So normally, if we take a tank and it's filled with algae and we have some shellfish, clams, or oysters in there, and we let them do their job, which is filter out water because they eat phytoplankton, that's their food, they'll eventually take a tank like this and clear it. They'll get rid of it. But this particular algae, Oreoumbra, which bloomed and is still up in the northern IRL, it produces a mucus on the outside of it. And that mucus can actually start to clog their filter feeding apparatus. Or the density can get so high they just can't feed effectively. So they'll essentially clam up, they can't feed, and they can starve and die. This was a really sad event. I actually was out in the field when this occurred. That little video I showed you, it happened from that organism, that particular dinoflagellate, Kashiwo, that is in the beginning of the talk. This animal, remember you could see it could swim, or this algae. It would swim up to the surface during the day and make these pretty, red, discolored red water on the surface. This produces a surfactant. Just like the Oreoumbra produces a mucus, this makes a surfactant that when seabirds would land in the water and this algae would all be up at the surface producing a lot of this organic surfactant, it would actually degrade the oil that keeps their feathers insulated. So seabirds, you see ducks on a frozen pond or in frozen water, they don't die because they're insulated by their oil. Well, this surfactant breaks down their oil. So the birds would land and they would suddenly get very wet and they couldn't dry off and they died of hypothermia. A lot of seabirds died during that time period. The last thing is economics that harmful algal blooms can do. This is, I don't know if I have to go into any great detail on this, but when you start to see signs like this is from the West Coast with the Florida red tide, or you see contaminated shellfish, this is from Martin County, avoid contact with the water. Don't do anything in the water. Don't drink the water. No one's going to do tourism in these areas, or it's going to drive the tourists away. Recreation, obviously, is out of the question in waters like this. You can't have fisheries. You can't take, shellfish and things from contaminated waters. So when these happen, a byproduct of all the other things they're doing, the economy can get seriously impacted. So the bigger issue is, and not to be more Dr. Doom, but here it goes. Worldwide, these HABs are increasing not only in geographical locations, but also in the frequency, duration, and severity of them. So if we look at these two maps, these are reported HAB occurrences around the United States. And this is old. If we were to update this, it would probably be quite different and more extreme than this. You can see a big difference between 1972 and 2008. And I believe that This increase in HABs is especially true for our Florida waters and the Indian River Lagoon. And I think if Florida is not number one, we're in the top 3 of one of the most impacted states in the United States. We have a serious harmful algal bloom problem here. And you would immediately say, why? Why do we have it? Why are HABs increasing? I'll go down a very quick list. You know, it's kind of like autism in the United States. People always ask, is it really getting worse, or are the doctors just getting better at diagnosing it? It's kind of like HABs too. Is it really getting worse, or do we just have better knowledge and detection of them? Now we see them. It's probably a little bit of this, but it's more of this. Eutrophication is a big problem, and this is simply nutrient pollution into our waters. Introducing new species to new environments. I don't know if you realize this, but big tankers, when they're overseas, they fill up with ballast water, local water. They come over here, they dump their ballast water, potentially. That ballast water has organisms or potentially harmful algal bloom phytoplankton that have never been introduced to these waters before, but when you turn them loose, they might find this a very nice environment to grow in. So we transport, like viruses around the world, harmful algal blooms. This is one mechanism for that. As humans, we do a lot of ecosystem modifications. We like to live by the water. We do things. We try to control the environment. We change, you know, we put canals in. We do dredging. We have aquaculture. All these things can change how the environment works and can change how the system functions. Again, climate change is definitely an issue. Whether you believe it's man-made or it's natural, it doesn't matter. It's happening. The warming is a huge problem because this increases the ranges and growth rates of harmful algae. Much like a lot of animals, you know, you don't find penguins living at the equator, usually. I mean, animals like different temperatures. And when it gets warmer, it allows different species to exploit an area where they couldn't before because the temperature was too low for them. And when it gets warmer, things grow faster. So those two things can create a significant problem. Along with climate change, we're having changing precipitation patterns. We get times of drought or times of very heavy rain and runoff. Increased runoff can carry nutrients and other things into our water. It's an issue. We also have more extreme events like, you know, stronger hurricanes. That can do serious damage and change to the local ecosystems. And the last one is we can change the primary grazers that exist in a system. And you can think of like oysters or clams. These are predators on these algae. They eat them. But if something happens, the clams die, or you know, something changes in the environment where these grazers are impacted, well, all of a sudden there's nothing to control the algae from growing. So now I'm going to give you a quick tour of what's going on in South Florida as far as the major HAB events. This is not the be-all, end-all presentation of everything that's going on, but this is the major stuff most of you probably have heard of. The first one is the one I've already mentioned, the Florida red tide. It's by another dinoflagellate. This is, again, one of these organisms that can swim, called Karenia brevis. It produces brevetoxin, which can give you nausea, vomiting, numbness, and respiratory distress, as I already mentioned. And the good thing about this one is we know a real lot about it, and we have an excellent monitoring system for it. USF, the FWIC, they have this constant report out there. You can go look at any time where Karenia is and how much it's blooming. I wish we had the kind of money that they did for monitoring the lagoon, for keeping track of this. This one is really well known and really well tracked in Florida. This other one, I can almost guarantee everyone's heard about, but this is Lake Okeechobee from space. And if you look, if you can see, hopefully it shows up. You can see all this green streaking on the surface. This is the blue-green algae blooms in Lake Okeechobee, which are recurrent. They've been happening for decades. You know, and this is the extent of this bloom. It's huge. I mean, this is gigantic. And if you look at it close to the surface, that's what it looks like. And this is from the particular cyanobacteria that blooms usually as the dominant. It's called Microcystis organosa. It produces microcystins, and these can cause liver damage or liver failure. So it produces a hepatotoxin. There are some people that think it can also cause neurological problems. And I should note that it's heat and very chemically stable, so cooking is not going to get rid of this toxin. Boiling water does not get rid of this toxin. And the chemical stability of it means that who's ever doing the drinking water out of a freshwater lake that has this, they've got to treat the water correctly, or it's going to go by, because it doesn't just come out easily like other components in the water. If you look at Lake Okeechobee, you can see that, besides the problem of the blooms that are potentially there, these problems can actually spread. If you look at the historic flow of how the lake used to be, the water would come down through the Kissimmee Water Basin, get into the lake, and essentially be shunted south into the Everglades system, south of the lake. Currently, it goes out into the Caloosahatchee River and through C-44 into the St. Lucie River and the St. Lucie Estuary. So it's going to take this algae with it when they have to discharge water out of the system. But it's not only discharging the algae, it's also discharging a lot of nutrients that are actually in the lake that come from this entire watershed. And they say, I don't know if this is correct or not, but 1/3 of the nitrogen that they measure down on this coast and coming out here, and nitrogen is something that can fertilize harmful algal blooms, is coming out of Lake Okeechobee. And you think about the big Corynea brevis blooms on this coast, well, this is probably helping feed them. I mean, we're dumping this nutrient out there, and we have the same problem over here. We're putting nutrients into our system. Another HAB in South Florida, how many people have heard of ciguatera? It's great. If you've lived in the tropics, you've probably heard of it. This is caused by another dinoflagellate, Gambardiscus. It tends to be epiphytic on algae and seagrass and other plants, so fish that pick at seagrass and eat it tend to ingest these at the same time. It produces ciguatoxins, which can give you nausea, vomiting, diarrhea, muscle pain, hallucinations. So if you eat a fish with ciguatera, you're going to have a bad night. It's also heat stable, so cooking does not get rid of it. It's another one of these toxins. And it's why ciguatera is not really a problem in the lagoon. This particular dinoflagellate likes warm water. The warmer the lagoon gets, we might start seeing Gambardiscus. I mean, Gambardiscus is here. We do have this species in the lagoon. Whether or not it can get to any density that will cause us a problem is another question. But overall, I keep track of a lot of these stories about how harmful algal blooms are affecting people. And recently they've said that, the cases of fish poisoning in Florida are a lot more prevalent than are reported because people they don't have a really bad, severe reaction. They just don't feel that good. So, you wonder how much people actually are getting exposed to this toxin. Another one in South Florida is Florida Bay. This is appears to be getting or worsening. So we have Florida Everglades, and there is some Lake O water that gets in there, and there is discharge out of the Everglades into Florida Bay. And this can be very nutrient rich. And it produces these high biomass blooms of cyanobacteria. This is not the cyanobacteria like Microcystis. This is actually a saltwater cyanobacteria dominated by Synechococcus. There's some pictures of these little tiny cells. And you can see a lot of these kind of reports now, these big algal blooms that are pretty equivalent to like our brown tide or the super bloom. It's very equivalent to what happened then, is now occurring a lot in Florida Bay. There's a big debate whether Synechococcus or some of these other cyanobacteria actually make any cyanotoxins. I don't know if there's been a definitive report. They might have microcystins or BMAA, which is a neurotoxin, and they could cause liver and neurological damage. But something like this is certainly going to cause anoxia and hypoxia. It's going to create fish kills, and it's going to kill any seagrass trying to make a living under there. So even though it's not toxic, it's one of these things that can be damaging. So what about our backyard? You all probably live around here. You're interested in what's going on in the IRL. So as I said, when I was saying what causes harmful algal blooms, we have continued development and changes in the land use practices around the whole system. It's a beautiful area. People want to live here. We have lots of development. Well, when we do this, we get an increase in point and non-point source pollution. We're changing the ecosystem and how nutrients get into it. When you do this, it leads to nutrient eutrophication, pollution, however you want to look at it, and very poor water quality. It's not nice and blue, and it doesn't look like beautiful. When you add it to those climate change effects I talked of, with increasing temperature and changing precipitation patterns and extreme events, you get an increase in HABs. This is kind of, it's not hard to see how it happens. And you can get an increase in the frequency, the duration, and where they occur and how often they occur. And just to bring this point home a little bit, the IRL is really big. It's somewhere around 140 miles or so. It's 40% of the Florida's east coast. It's so big, it spans 2 climate zones, subtropical and tropical. Unfortunately, the IRL is becoming more tropical in climate. It's getting warmer. Now, you don't have to take my word for it. The USDA has these plant hardiness zones. So they use to grow various plants, probably keep track of these. And you can see back 1994 to 86, this was a hardiness zone. You don't really have to know what they are. But the more red and yellow it is, the warmer it is, and the more hardy or the more tropical plants that can exist there. And you can see now, and this is outdated, this is from 2005, it's already got well up into the IRL now. This tropicalization, this turning it more totally tropical instead of subtropical, it allows these new HAB species to exploit the IRL. So it is a problem. Now we have had, harmful algal blooms will always occur. They occur everywhere naturally. Algae grow. The ocean's filled with algae. And we've had, since science has been looking in the IRL, there have been harmful algal blooms. But we have noticed, this is kind of a record of what occurred in before 2000, and some of them are into the 2000s. And it does appear like, well, it seems like something has changed. We're seeing different things exploit and take over the IRL, so to speak, in these blooms. This is the Microcystis organosa, the blue-green algae from Lake O. You can see these are some of the major reported events that have occurred. They're starting to happen frequently, every 2 to 3 years. 2011, we had the super bloom. That was cyanobacteria, not this cyanobacteria, Synechococcus and Pico eukaryotes. The brown tide is Oreoumbra lagranensis. That is still ongoing. And this is very different from what was up here. And this is why you could say, well, maybe that hardiness change or that warming of the lagoon is starting to allow these things to exploit it better. It's not, a definitive fact, but it's one possibility. And I'll give a shout out to Paul Hargraves, who I think is in the audience. Raise your hand. There he is. He's over there. He recently put a list together, and there are nearly 70 HAB species recorded of phytoplankton in the IRL. So any one of those at any time could potentially learn or exploit the conditions we're creating there. So think about how many that is. There's a lot of things that could potentially bloom in the lagoon. So what are the current HAB threats to the IRL? It's pretty much everything I outlined to you. We have hypoxia and anoxia, low and no oxygen. We've had fish kills. There's fish kills almost every year. Some are large, some are small. We have high light absorption, which causes shading and death of seagrass from these large biomass events. We have this exopolymer secretion or mucus from some of these species, which can affect the filter feeders and the shellfish, oysters, clams, etc. We obviously have economic problems. During 2016, Martin County had to declare a state of emergency, and their economy was seriously affected. And we have toxins. These are the 3 major ones that we measure in the IRL: saxitoxin, microcystin, and domoic acid. And these directly impact human and wildlife health. And for these 3 toxins, saxitoxins are very strong neurotoxins, very dangerous, causes paralytic shellfish poisoning. Domoic acid is another neurotoxin. It causes amnesic shellfish poisoning. And microcystin, as I talked about, this is a really strong hepatotoxin. It can cause liver damage and liver failure. So just to go over some recent ongoing HABs that are occurring right now or have recently occurred in the IRL and some of their effects, just to hammer this point home. I told you about the brown tide. It causes hypoxia and anoxia, fish kills, seagrass loss. In 2016, I mentioned this large fish kill. We went up there, I think, 2 days after the fish kill, right when the bloom was really, really strong. And we took some whole water and we put it under a microscope and imaged the cells. And all these this is whole water. It's not concentrated or anything. It's a very, very tiny amount of water. All these little dots you see, bright dots, are all individual Oreoumbra cells. So they're about 4 microns. They're very, very small. You cannot see them with your eye. We recently went up there in December, just a few months ago, and this is what the whole water looked like. We saw as many as we did during the 2016 fish kill. There were millions of cells per milliliter. That's not to say there is a fish killer. There's going to be low oxygen, but when the water warms up, I fear we could see some kind of repeat of this. The biomass is there. This bloom is still very healthy and still going on. And it's interesting. I put this little title from Ed Buskey, is a colleague of mine from Texas. This same exact event happened in Laguna Madre. They had a persistent, continuous 10-year bloom of Oreoumbra in Laguna Madre. And we could be looking at the same thing. It started in 2012. It could go for many, many years. So we have that. The next one I'll go through is we have this pyridinium red tide. I call any dinoflagellate bloom a red tide. I don't like that they co-opted the name for the Florida red tide. It's what the old school people, anything that was a dinoflagellate, we call it a red tide. This pyridinium, which occurs up in the Banana River in the NIRL, it produces this saxitoxin, this very dangerous neurotoxin. It can give you paralytic shellfish poisoning. So if we look at it, the postdoc in my lab, Malcolm McFarland, who is definitely here somewhere, isolated it. We got it in culture, and this is the pyridinium swimming around. Looks very happy. You can see it's another dinoflagellate. It's modal. It can move around. And this is a recurrent summertime bloom since the mid-90s. This thing has been going on for quite a while. And I say summertime in quotes because, I mean, summertime here is kind of all year. But it goes probably roughly from May to maybe November, somewhere, and plus or minus a few months, depending on the temperature. It likes warm water. It produces this saxitoxin, as I said. It led to a permanent ban on taking pufferfish from the lagoon because it concentrates in pufferfish, apparently. And we had a really strong bloom this year. It was really strong. There were high cell numbers, and it was over a very large extent of the northern IRL. And we measured the toxin in this, and we went out and did it, and we were measuring between 3 to 8 micrograms per liter saxitoxin in the water at times. And you say, well, what the heck's that mean? And to me, just to give you an idea of what that actually means, again, we measured somewhere between 3 to 8 micrograms per liter. These are the EPA drinking water guidelines. I found them, I just pulled it out for a couple states. We actually don't have one of these for Florida. I'm sure it's somewhere. But if you look at Ohio, and this is drinking water guidance and the action level, there's 3 things here. Do not drink children under 6, saxitoxin 0.2 micrograms per liter. Do not drink children 6 and older and adults, saxitoxin 0.1. So it's not good for anyone. Don't drink water with saxitoxin in it. Salt water, we don't have to worry about it. This is kind of the disturbing one. Do not use, based on recreational, no contact, recreational closure. Don't go in the water. Saxitoxin, the level for this is 3 micrograms per liter. Seems suspiciously close to what we measured. And it's funny. I was on Florida today, not that long ago. I think it came up on my Facebook feed. And it was telling me, and I'm new to Florida, authentic Florida, check out these 18 discoveries to add to your bucket list. It's like, oh, awesome. It says, see the mesmerizing lights up in the northern Indian River Lagoon by Merritt Island, etc. Who's heard of these bioluminescent kayak tours? Yeah, a few people. Well, anyway, USA Today is pimping it and saying we should do it. Well, guess what? Pyridinium is a species that creates this bioluminescent. It happens to be a bioluminescent dinoflagellate. Pyridinium is toxic. Lots of bioluminescence means lots of toxin. They're telling you to tour in toxic water. So it's not a bucket list. It might be a kick the bucket list. I'm not sure. And I find this very ironic. It's like, well, whatever. Now, as I said, Pyridinium bahamense is a summertime bloom in the IRL, and there it is again. And you'd probably ask, where does it go in the winter? What does this thing do? Dinoflagellates are really cool organisms. Like many dinoflagellates, when it gets into a situation that it doesn't like, low temperature, or it runs out of nutrients, or whatever, the environment turns in a way that it doesn't like it, it actually produces what are called cysts. So this swimming organism turns into this. This isn't modal. It just sinks to the bottom and gets into the mud or the sand or whatever. And if you look at this simple diagram of how this works, these cysts will eventually hatch when conditions turn favorable. They'll start swimming and doing their thing, and then when things turn bad again, they go through a sexual life cycle and they produce more cysts. And here's an actually, it shows one of these actually hatching, which is kind of cool. So this is what it does. It will produce cysts, it overwinters, and then it hatches the next spring, summer. So we get through that. At the same time we were up doing this pyridinium sampling, we also found a pretty large bloom of Pseudonychia. Pseudonychia is a whole bunch of different species of diatoms. Some are toxic, many of them are not toxic. And there were a lot of them up there. And we didn't know if it was a toxic version or not. You can't tell by, it's very hard, you can't tell by looking at them, essentially. So we started testing for domoic acid. And sure enough, there was some toxic species in there. It was a toxic Pseudonychia bloom. We detected it in the Banana River. So we know there are now definitely toxic species that are blooming. And it's interesting too, this has been a long-standing problem on the west coast of the United States. But back in my old hometown in Rhode Island, they recently, over the past few years, have started seeing blooms of this, where they really didn't see these large blooms before, and it apparently came back again. And I start to fear, has it got a foothold on the East Coast now for some reason? And not that these are related, but it's funny that Rhode Island had a problem these past 2 years, and now we've just seen some here. So this bears watching what's going to happen with it. Another one that happened along most of the northern IRL, and this actually extends way down. I kind of restricted that. And this is always a background in the IRL. You always have all this cyanobacteria and other phytoplankton that are growing. And sometimes they get really high in biomass, and sometimes they're just the low-level background that's there. But they're almost always there. But eventually, they are what caused the super bloom. So in 2011, if you were here, when it happened, it killed a lot of seagrass and was a serious problem. Hypoxia, anoxia, fish kills, seagrass loss. All that happened, and this stuff's always there. It's just whether or not you're going to have this recurrent event where it's going to get really bad. The last one, I talked about this a little bit before, but we have Microcystis coming out of Lake O, getting into the St. Lucie Estuary and the IRL, and actually getting to Atlantic beaches as well. We call that a green tide. It produces, again, these microcystins, which are a liver toxin. It can also, because there's so much of it, it can cause hypoxia, anoxia, fish kills, and all the other things. So these are all the things that are pretty recent and going on in the IRL. And just to go a little bit more into this Microcystis green tide, this has been happening every several years for decades. This is not a new phenomenon. It does appear to be getting worse. Lake Erie is seeing more extreme blooms of it for whatever reason. Microcystis, and a lot of people are saying worldwide now, microcystis blooms are starting to just become more prevalent in fresh water. So it comes out, as I said, of Canal 44, it gets into the estuary, and it produces these liver toxins. Well, now I'm really going to be Dr. Doom. This was, I need the green suit now. This was a 2017 Associated Press report out of the TC Palm, and it also got picked up by Health News Florida. 4 counties part of liver disease and algal bloom cluster. 4 counties along Florida's Treasure Coast make up a cluster with high rates of deaths from liver disease and algal blooms. What are those counties? Indian River, Martin, St. Lucie, and Okeechobee. It's the only one in the state of Florida that clustered out like this. I went and dug up this paper. This is the actual research paper right here. And it's non-alcoholic liver disease, so we can't blame it on that people really like to drink in those 4 counties. And this is a map. It's kind of the crux of this paper. Wherever it's red, that's bad. That's where there's a correlation between these cyanobacterial algal blooms and liver disease and death. And those are the 4 counties in Florida. I note 2 other ones because I have some experience with them. San Francisco Bay has a very similar problem to the St. Lucie Estuary. There are large microcystis blooms which eventually lead out into San Francisco Bay. And it's kind of a recurrent problem there. And sure enough, they clustered out one of these areas too. And Lake Erie, I've already talked to you about that. If you don't know, this is Lake Erie, Great Lakes. And the blooms are always right in here. And they clustered out there. Now that looks tiny, but that area has millions of people in it. So it's a problem. So what are we doing? I mean, this is the thing. I can try to scare you all day, but eventually we're going to use science and try to, somehow fix or mitigate this problem. So what are we doing? I've approached NOAA last year. The NCCOS gave us funding to look at the brown tide and start looking at some of its ecology. At the same time, we were funded to look at this cyanobacteria and these phytoplankton that are kind of background growing here. The Harbor Branch Foundation, through the Protect Wild Dolphins Specialty License Plate funding, supported us to look at this pyridinium red tide. So we're currently up there measuring the toxin in the water. We're measuring toxin in the prey fish of dolphins and in dolphins themselves, if we can get them from strandings. just to see how is this toxin getting into the food chain. And the dolphins are like sentinels for us. If it gets into the dolphins, there's a good chance it's probably getting into us. We eat some of the same fish they do. And lastly, for the Microcystis bloom, the green tide, the Everglades Foundation also gave us funding to start tracking toxins of this in the system, although that funding ran out, unfortunately. We're currently still working. We have 2 efforts going in through the NEP, the National Estuary Program. I have a collaborative effort with Ed Flips, who does a lot of sampling for harmful algae in the lagoon. We want to look at better monitor just all the species across the whole lagoon. Ed tends to only look in the northern area. We're also thinking about applying to do some better monitoring down in the south with Dennis. We already kind of have a program going, but we want to make it official. Hopefully we'll get some traction with those. And we also want to do this. I'm currently working with NASA. We want to establish what's called a NASA AERONET OC site, which stands for Ocean Color, in Lake Okeechobee, and we want to start this like in the next month or 2. And this is a collaboration with NASA and a colleague of mine at UNH and the South Florida Water Management District. And we want to put out this sensor, which essentially looks down, and it's going to be on LZ40 water tower, the South Florida Water Management District's water tower, water quality tower. It looks down at the water every hour and tells you essentially how much algae is there. So you can constantly track it instead of having to send someone out there weekly or monthly or try to get a good satellite image. It's constantly monitoring what's there by looking at the color of the water that reflectance is coming up. And as I think Dennis alluded to, this project actually, hot off the press, this was just in the paper today, that we are actively trying to do this. Hopefully it'll all get worked out. But this is going to get us yet another data point for us to understand how these blooms, what makes them wax and wane, be strong or be weak in the lake. And maybe we can use that to say, now's not a good time to discharge water. There's a little bit too much there. So I say my vision, but it's not my vision. It's a whole bunch of people's vision that I talk to and work with and collaborate with, is that we have this coordinated spatial temporal mapping, monitoring, and data assimilation to inform management and policy decisions, to inform the public if there's danger, to figure out if our mitigation, if our dredging, are all those things working. And we need to take all the assets we have, the satellite, drones, the LOBO sensors, or Orca, or all the different ways that we can get data, our surface sampling from boats, autonomous devices, and start integrating that with everyone. All these people are out there, and we kind of coordinate, but we don't really have a central way of coordinating this kind of research. And there's a lot of data we have to put together. One of the ways that we're going to try to do this, and it's a big challenge. If you think about it, if we're going to understand this, we're going to understand these HABs and these dynamics, and I just showed you how complicated it is. It's not looking at one thing, it's looking at many things. Where these toxins are going in the water, how they're getting in the food chain, and how they're affecting us. That one study was just for microcystin. It didn't track any other toxins. We don't know what the other toxins are really doing. And how we're going to manage this. It requires an effort between scientists, the public, water management, policymakers. I mean, we all have to get involved. One of the solutions we have is that we're trying to create a Center for Oceans and Human Health here. We have a pending proposal to the NIH to essentially do what I just told you. And we're going to know about this within the next week or 2. If it doesn't get funded, I'll just keep on keeping on and try to find money elsewhere. But we're working on it. We're trying to do better. And with that, I will take your questions.